Right, guys. So there are two important terms which you have to understand, guys. For example, if you are using a method to kill all the organisms, right? If you are using a method to kill all the organisms, all of them. So that method, what do you call it as? So what did I tell you? I told you that if you are destroying all forms of organisms, right? If there is destruction of all forms of organisms, if there is destruction of all forms of organisms, so you call that condition as what? Sterilization. You call that as sterilization. For example, if you are destroying all organisms except the spores, right? So if there is destruction of all forms of organisms except spores, you call this thing as disinfection. So right now what we shall be doing is that we shall be discussing two main topics. One is called as sterilization, and the other one is called as disinfection. Sterilization as well as disinfection. These are the two important topics which we will be discussing right now. Okay, so I hope you understood what is sterilization and what is disinfection. So what are the different methods of sterilization? How are you gonna sterilize? For example, if there is an let us say OT room, right? Operation theater. How are you gonna sterilize the operation theater? How are you gonna let us say sterilize the surgical instruments? So we have to discuss the methods. So what are the methods of sterilization and disinfection? What are the different methods of sterilization and disinfection which you basically use? So there are two methods of sterilization as well as disinfection. Now what are these two methods? One is called as a physical method. Another one is called as the chemical method. So what are the two methods? One is called as a physical method and another one is called as a chemical method. Now chemical method is you use the chemicals and completely sterilize that area. Okay, you use the spirit, you use the betadine and remaining things and sterilize that area completely. But when it comes to the physical methods, what are the different kinds of physical methods do we basically use? What are the different kinds of physical methods do we basically use? So there are three physical methods through which you can kill all the organisms. All of you know the first thing, that is heat it, right? So heat is a physical method. Apart from heat, second thing is you can filter the microorganisms, filtration, heat, filtration, and third important thing is radiation. Okay, so what we have discussed, guys, there are two methods of sterilization and disinfection. One is called as a physical method and the next one is called as your chemical method. Okay, when it comes to the physical method, you can see heat, filtration as well as radiation. So we shall be discussing what forms of heat are available. So through heat, within this heat there are many methods. Okay, and within this filtration there are methods, within this radiation also there are methods. Okay, so we shall be discussing first regarding the heat. So through heat, through what temperatures, what is the timing, how much time you gonna heat, right? So everything we shall be discussing regarding the heat. So through heat, how are you going to destroy the microorganisms, how are you going to sterilize and disinfect, we shall be discussing right now. So let me make a table of that. So if you are heating anything, right, if you are heating anything, you can heat by two ways. because there are two ways of heating. What are how many ways? There are two ways of heating. Okay, so one way you call it as dry heat. Okay, so one is called as dry heat. Now when I tell dry heat, the opposite of dry will be moist. So obviously there will be moist heat. So guys, there are two types of heat which you have learned. One is called as dry heat and the other one is called as your moist heat. Okay, fine. So through dry heat, how am I going to destroy the microorganism? So through dry heat, within this dry heat, there are three ways how you can destroy the microorganisms. The first way is called as flaming. You can directly put the flame. There are different degrees like 80 degrees, 180 degrees, right? So at different degrees, the microorganisms are gonna die. So flaming is one method. Flaming. And after flaming, the next important thing is incineration. The next important thing is incineration. So what is incineration? I will be telling you, but just for now, believe me that there are three methods in dry heat. One is called as flaming, another one is called as incineration, and the third one is called as hot air oven. Hot air oven. Okay, but when it comes to moist heat, there are many different ways, guys, many different ways. So first of all, there are three different ways. What are the three different ways? Are one is there are some methods where you provide moist heat with a temperature less than 100 degrees centigrade. There are some methods through which you can provide moist heat and kill the organism with a temperature of approximately 100 degrees centigrade. There are some methods through which you can provide moist heat and kill the organisms at a temperature of, you are providing the temperature of moist heat of more than 100 degrees. So in one scenario, you are providing moist heat of less than 100 degrees and killing the microorganisms. You are providing equivalent to 100 degrees and you are providing more than 100 degrees temperature of moist heat to kill the microorganisms. So within this less than 100 degrees part, what all the methods are there? For example, the first, let me write it like this: 1, 2, 3 and 4. Now the first important thing is that pasteurization. Pasteurization, mainly we use this technique for milk, right? So in the milk, if you want the milk to store for a long time, if you want to increase the shelf life of the milk for a long time, what you do is that you kill the microorganisms in the milk, right? So for that you need not to use more than 100 degrees. You can just use less than 100 degrees. Okay, less than 100 degrees of moist heat is enough. Next one is called as serum bath. And third one is called as vaccine bath. And the last one is called as inspication. So these are the four ways by which in which you use temperature of less than 100 degrees and kill the microorganisms. Okay, so what type of heat you are providing here? Obviously less than 100 degrees, but what type of heat? That is moist heat. Now in what all conditions you provide a heat of equivalent to 100 degrees? So equivalent to 100 degrees, you provide in only one thing. That method is, so what is that method, guys? That is called as tyndallization. And finally, you used moist heat of more than 100 degrees and by a method called as autoclave. So this is a general, I can tell you, a general bird view of what we are discussing. regarding the heat. So our main topic today will be regarding the heat. So within the heat there are two types. One is called as dry heat and moist heat. Dry heat you do by three methods. One is called as flaming, incineration and hot air oven. Next, moist heat you do by three ways. You provide three different temperatures. One is less than 100, equal to 100, more than 100. Less than 100, three methods. One is called as pasteurization, serum bath, vaccine bath, two baths, right? Pasteurization and inspissation. Next one is equivalent to 100 degrees, that is sterilization, and finally you have got your autoclave. So guys, I told you about the dry heat and the moist heat, right? So obviously as there are different methods in case of dry heat, as I have mentioned, as well as there are different methods in case of moist heat as well, right? So moist heat and dry heat, you have got the different methods, but the important thing to discuss here is that how in dry heat are you going to kill the microorganisms, and how in moist heat, when you provide moist heat to something, right, to let us say milk or something, how are you gonna kill the microorganisms, how the microorganisms are killed within that milk or whatever it is when you are providing moist heat. So when it is dry heat, there are three ways by which the microorganism is killed, right? I'm talking about the mechanism, okay? So that mechanism is applicable in all these three things. In flaming also, the microorganisms will die by the same way. In incineration and hot air oven also, it will die by the same way. So what are these methods are? The mechanism of action is, for example, let us say I have taken scissors or something, okay, some kind of surgical instrument. Now I am providing dry heat to that so that the microorganisms on that surgical instrument, let us say scissors or scalpel, so they have to be completely dead, they have to be killed completely. Okay, so what will happen is that first thing is, whenever I am providing dry heat, within that microorganisms, three things can happen. For example, if I am a microorganism, you are heating me so that I have to die. So three things happen in my body. What are those three things are? Whatever proteins are there in my body, they will be denaturated first. So what will happen? Denaturation of proteins. Denaturation of proteins. And second thing is, when you are heating me, the electrolytes which are there in my body, they are undergoing imbalance, right? So there is electrolyte imbalance. Electrolyte imbalance, and finally there will be oxidative damage. Oxidative damage. So these are the three ways by which in dry heat you are able to kill the organisms. But when it comes to the moist heat, how are you going to kill the microorganisms? Is that the first important thing is obviously denaturation. Denaturation of what? Denaturation of your proteins. Okay, denaturation of your proteins. Second thing is that in moist heat you are providing not dry heat, you are providing moist heat, right? So in moist heat, electrolyte imbalance, oxidative damage are not seen, but what is seen is that there will be coagulation of proteins. There will be coagulation of protein, there will be denaturation as well as coagulation of proteins. So these are some of the important things to understand: denaturation, electrolyte imbalance and oxidative damage, and here denaturation of proteins and coagulation. So these are what? These are the mechanism of actions by which the respective heats, whatever it is, dry heat or moist heat, and these are the mechanism of actions for these respective heats. Okay, now we shall discuss mainly about the dry heat. Within the dry heat, the first thing is flaming. Okay, so what did I tell you? We shall be discussing what? We shall be discussing a topic of dry heat. I hope you understood what are the mechanisms by which within the dry heat the microorganisms are killed, and within this dry heat we shall be mainly focusing on what? Flaming. So simple thing guys, anyways there are many temperatures to remember. I'll write down all of them at the end, but as the topic is going on I have to write it subsequently here. So flaming in the sense, right, flaming in the sense you have to heat the you have to heat a specific, let us say any lab instrument till it gets red hot. For example, I hope all of you know what is inoculation loop. Inoculation loop and wire. I hope all of you know what is inoculation loop and wire. So in the lab you might be seeing this. So you will see this inoculation loop like this, isn't it? So what you do guys, you take a petri dish, you move the microorganisms with this, or you transfer microorganisms from one plate to another plate because of with the help of this inoculation loop and wire, right? So obviously you cannot use it without sterilizing it, right? So what you do is that you have to heat it. So how do you heat it? You put it on the flame. I hope you have seen it many times that you are putting it on the flame to heat that microorganisms. Mainly you place this part because this is the part which is in always contact with the microorganisms. So you heat it to red hot, right? So as you are flaming it, when you are putting it on the flame, slowly the color will turn to red hot. Okay, so when it is turning to red hot, you can think of three things, right? So within this flaming we basically use three different temperatures. One is 800 degrees centigrade temperature, another one is 1200 degrees centigrade temperature, and the other one is 400 degrees centigrade temperature. So out of these three, 1200 degrees centigrade is the most commonly used temperature. Most commonly used temperature to heat this particular inoculation loop or wire. So very simple. Within this flaming, for flaming, what is the material you use is that basically you use the inoculation loop and wire, right, to kill the microorganisms on that. Clear? How do you do it? By three temperatures, 4, right, 8 and 12. Clear? So 400 degrees, 1200 degrees and 800 degrees, and 1200 degrees is the most commonly used one. Clear? So after this we are done with the flaming part. Now we are going with incineration. Now we shall be discussing about incineration. What is your concept of incineration? What is this incineration? Incineration means you can burn. I mean, incineration is a method of dry heat where you burn anything to ashes. So if you are burning it to ashes, burn it to ashes. Ashes, why are you burning it to ashes? You only burn those things which are not at all useful for you, right? So what is that medical equipment, what is that medical waste which is not at all useful for you guys? Just think. For example, in the biomedical waste there are three colors, in which one of them is yellow color bag. Within that yellow color bag, what do you throw? Sharpeners. You don't throw sharpeners onto that. What do you throw is that you throw the dressing materials, you throw bandages, you throw swipes, right? Whatever swab material is there, that thing you throw. And iodine. So iodinated materials, whatever is there, that thing also you do. So what you do is that early morning you go for the rounds, you have to change the dressing of the patient. So you will remove the previous dressing, and what do? You put that in a yellow color bag and you have to completely burn it into ashes. Okay, because they are highly infectious and contagious. Clear? Because there is a lot of patient blood mixture fluids over there, so you have to completely heat, you have to burn it into ashes. So for that you have to use this method called as incineration. So if you have to burn something into ashes, Don't you think the temperature which you are using should be too high? Isn't it? The temperature which you have to use has to be too high. So the temperature which I basically use here is 1000 degrees. 1000 degrees centigrade to 1200 degrees centigrade. So this is the basic temperature which I use. 1000 degrees to 1200 degrees centigrade. So what are the materials which I use here? What are the materials which I use here are the biomedical waste. Biomedical waste, all the things which are located in your yellow bag, everything that is in your yellow bag, right? So what are the things which you find in your yellow bag? What are the things which you find in your yellow bag, guys? So within your yellow bag you have got your dressing materials, right? You have got your swabs, you have got your wipes, right? You have also got your let us say iodine, right? So all of these are highly infectious clinical waste. So all these things you have to just burn it into ashes.